Photosynthesis, using light to make food. Nearly all life on Earth, including you, can trace its source of energy back to the sun. Photoautotrophs feed us, clothe us as a source of cotton fibers, house us in the form of wood, provide energy for warmth, and provides transportation as biofuels. Specific wavelengths of visible sunlight, carbon dioxide, and water are the requirements for photosynthesis. The products of photosynthesis are oxygen, glyceraldehyde 3-phosphate, or G3P, as well as simple carbohydrate molecules that can be converted into glucose, sucrose, or any of dozens of other sugar molecules. Organisms that are able to undergo photosynthesis include plants, algae, and cyanobacteria, which is a group of bacteria. Photoautotrophs literally translates into self-feeders using light. Photoautotrophs use light to manufacture their own food. You can see in the photosynthesis equation that 6 carbon dioxide molecules plus 6 water molecules in the presence of sunlight will create 1 glucose molecule and 6 oxygen molecules. Heterotrophs translates as other feeders. Heterotrophs must rely on the sugars produced by photosynthetic organisms for their energy needs. This includes animals, fungi, and most other bacteria. Chemoautotrophs feed on inorganic chemical compounds. Chemoautotrophs synthesize their sugars by extracting energy from inorganic chemical compounds. For example, there are nitrogen-fixing bacteria and iron-oxidizing bacteria. Chloroplasts are the sites of photosynthesis. All green parts of a plant have chloroplasts. Their green color is from chlorophyll. Chlorophyll is a pigment which is a light-absorbing molecule that exists in the chloroplast. It plays a central role in converting solar energy into chemical energy. The process of photosynthesis occurs in the middle layer of the leaves in an area called the mesophyll. Openings located on the underside of leaves, called stomata, allows for gas exchange. Carbon dioxide goes into the plant for photosynthesis, and oxygen is released. Chloroplasts have a unique structure which includes a double membrane. Within the double membrane, thylakoids are stacked, disc-shaped structures located within the chloroplasts. The thylakoid lumen is an internal space enclosed by the thylakoid membrane. A granum is a stack of thylakoids, and the stroma is the liquid-filled space surrounding the granum. Chlorophyll is a pigment molecule that absorbs light. It is embedded in the thylakoid membrane and is responsible for the initial interaction between light and plant material. Photosynthesis takes place in 2 sequential stages. The first stage is called the light-dependent reactions. The second stage is called the light-independent reactions, or the Calvin cycle. In the light-dependent reactions, energy from sunlight is absorbed by chlorophyll, and that energy is converted into stored chemical energy. In the light-independent reactions, or Calvin cycle, the light-dependent reactions drives the assembly of sugar molecules from carbon dioxide. The light-dependent reactions utilize certain molecules to temporarily store the energy. These are referred to as energy carriers. The energy carriers that move energy from light-dependent reactions to light-independent reactions can be thought of as full because they are rich in energy. After the energy is released, the empty energy carriers then return to the light-dependent reaction to obtain more energy. The light-independent reactions of Calvin's cycle are called light-independent because they do not use light as a reactant, but they are not truly light-independent. This process uses energy made in the light-dependent reactions. Many of the enzymes needed for these processes are activated by the sunlight. The sun emits an enormous amount of electromagnetic radiation. Humans can only see a tiny fraction of this energy, which we refer to as visible light. Our retinal pigments can only see or absorb wavelengths between 700 nanometers and 400 nanometers of light. Thus, this area of the electromagnetic spectrum is called visible light. The manner in which solar energy travels is described as waves. The amount of energy of a wave depends on its wavelength. Shorter wavelengths are more powerful than longer wavelengths. The distance between consecutive crest points of a wave is considered a wavelength. A single wave is measured from two consecutive points, such as from crest to crest or trough to trough. Light energy initiates the process of photosynthesis when pigments absorb specific wavelengths of visible light. Light energy excites electrons. When a photon of light energy interacts with an electron, the electron may absorb the energy and jump from its lowest energy level, which is the ground state, to an excited level. Chlorophyll A and chlorophyll B are pigments that are found in chloroplasts. In the light-dependent reactions, solar energy is converted into chemical energy in the form of NADPH and ATP. NADPH and ATP supports the light-independent reactions and fuels the assembly of sugar molecules. A photosystem consists of 1. A light harvesting complex, and 2. A reaction center. Pigments in the light harvesting complex pass light energy to 2 special chlorophyll A molecules in the reaction center. The light excites an electron from the chlorophyll A pair, which is then passed to the primary electron acceptor. The excited electron must then be replaced. The actual step that converts light energy into chemical energy takes place in a multi-protein complex called a photosystem. 2 types of photosystems are found embedded in the thylakoid membrane. These are photosystem 2 and photosystem 1. Light energy is converted into an excited electron. The reaction center contains a pair of chlorophyll A molecules that undergo oxidation upon excitation. All of the subsequent steps involve getting that electron onto the energy carrier, the NADPH. This is done for delivery to the Calvin cycle, where the electron is then deposited onto carbon for long-term storage in the form of a carbohydrate. Photosystems 2 and 1 are 2 major components of the photosynthetic electron transport chain, which also includes the cytochrome complex. The cytochrome complex transfers the electrons from the carrier molecule, plastoquinone, to the protein, plastocyanin. This enables 2 things. First, it enables the transfer of protons across the thylakoid membrane. And 2, it enables the transfer of electrons from photosystem 2 to photosystem 1. In the photosystem 2 reaction center, energy from sunlight is used to extract electrons from water. These electrons travel through the chloroplast electron transport chain to photosystem 1, which reduces NADP+ to NADPH. The electron transport chain moves protons across the thylakoid membrane into the lumen. At the same time, splitting of water adds protons to the lumen, and reduction of NADPH removes protons from the stroma. The net result is a low pH in the thylakoid lumen and a high pH in the stroma. ATP synthase uses this electrochemical gradient to make ATP. In plants, carbon dioxide, or CO2, enters the leaves through holes called stomata. The carbon dioxide diffuses through the stomata over short distances through the intercellular spaces until it reaches mesophyll cells. Once in the mesophyll cells, the carbon dioxide molecules diffuse into the stroma of the chloroplasts. Recall that the stroma of the chloroplast is the site of the light-independent reactions of photosynthesis. The Calvin cycle is named for the man who discovered it, and because these reactions function as a cycle. The light-independent reactions of the Calvin cycle can be organized into 3 basic stages. 1. Fixation. 2. Reduction. 3. Regeneration. Stage 1. Fixation. In the stroma, the light-dependent reactions are initiated by 3 carbon dioxide molecules, 1 Rubisco molecule, and 3 RuBP molecules. Rubisco is the abbreviation for ribulose 1,5-biphosphate carboxylase oxygenase. RuBP is ribulose biphosphate. Rubisco catalyzes a reaction between carbon dioxide and RuBP. For each carbon dioxide molecule that reacts with 1 RuBP, 2 molecules of 3-PGA, or phosphoglyceric acid, form. The number of carbon atoms remains the same. As the atoms move to form new bonds during the reactions, the number of carbon atoms does not change. 3 carbon atoms from 3 carbon dioxide molecules, 15 carbon atoms from 3 molecules of RuBP, equals 18 carbon atoms in 6 molecules of 3-PGA. This process is called carbon fixation because carbon dioxide is fixed from an inorganic form into organic molecules. ATP and NADPH are used to convert the 6 molecules of 3-PGA into 6 molecules of glyceraldehyde 3-phosphate, or G3P. This is a reduction reaction because it involves the gain of electrons by 3-PGA. Recall that a reduction reaction is the gain of electrons by an atom or molecule. Stage 2 is reduction. Here, 6 molecules of both ATP and NADPH are used. ATP is hydrolyzed to form ADP and energy is released. NADPH is reduced, forming NADP+. Energy is released and a hydrogen atom is lost. ADP and NADP+ return to the nearby light-dependent reactions to then be reused and re-energized. Stage 3, regeneration. One of the G3P molecules leaves the Calvin cycle and is sent to the cytoplasm to contribute to the formation of other compounds needed by the plant. The remaining 5 G3P molecules remain in the cycle and are used to regenerate RuBP, which enables the system to prepare for more carbon dioxide to be fixed. 3 molecules of ATP are used in these regeneration reactions. Photosynthesis consumes carbon dioxide and produces oxygen. In contrast, aerobic respiration consumes oxygen and produces carbon dioxide. These 2 processes play an important role in the carbon cycle. Thank you for watching.